hi, a very happy good evening. So welcome to my session. So today we are talking about biology, the story of life. So today we are going to talk about first fertilization events. For all those who are new to my session, so Monday, Wednesday, Friday I teach class 12 portion, and Tuesday, Thursday and Saturday I teach a class 11 portion. So I was teaching. So today we are here talking about the last topic under your sexual reproduction and flowering plants. So today we are going to say a goodbye to this chapter. So we're almost done with this chapter, and I guess you guys remember tomorrow there is a mentee, so please do join for my mentee. So depending on tomorrow's mentee, so I'll decide whether I have to conduct further mentees or not. Okay. Yes, so what we'll be talking in this chapter, so sexual reproduction and flowering plants is a very important chapter from your term 1 point of view as well as from your NEET point of view. It's a very important chapter and always you tend to get questions from here. So maximum you have 3 questions. So 3 questions, so right answer gives you 4 marks. So you have 12 marks coming from this chapter. Hi Sneha, Sunil, a very happy good evening. So thank you so much for joining my session today. So please do like, share and subscribe. So let's move forward. So we're going to talk about post-fertilization events, and guys, once again it's a gentle reminder, so tomorrow at 5:15 p.m. so we are having a mentee. Same time tomorrow we are going to have a mentee, so we are going to play for 25 questions. So the topic is neural control and coordination, so please join me for the same. Alright, so please share the link with your friends, so let your friends know as well. So more the number of participants, more fun it is. For all those who are new, mentee quiz is a type of quiz that we play, but you will open it in your mentee.com. So I will give you a code, so when you log into the code you can see my questions. So the person who answers quickly and who answers rightly fetches more marks. So it is like a game. Alright, fine. So let's move forward. So Amarnath, hello, hi. So thank you for joining my session. So guys, please do like, share and subscribe. So let's move forward. Recap. So before we go to the recap, yes, Amarnath, good evening. So Sneha and Amarnath, can you give me a quick survey as to what topics in class 12 is tough for you? What topics in class 12 are tough for you? Because I have seen, so Sneha and Amarnath come for only class 12 topics. So in class 12, what are the topics that is tough for you? So can you quickly give me the topics that are tough for you? The might be the chapter name also you can say, might be the topics also you can say, but can you tell me which are the tough zones for you so that I can address them? Genetics, yes, okay. Fine. Apart from genetics, Sneha says genetics, okay, because instead of going chapter wise, I would only go for important topics because your term 1 exam is also coming for CBSE. So I thought I'll quickly address your important topics. Okay, so now today is the last lecture for your sexual reproduction. So next I will teach genetics, so that will be easy for you. So next week I have planned only 3 days classes. So 3 days I will make only important topics to you. Okay, so apart from genetics, any other topic in class 11 that is tough for you? Yeah, genetics I will do fully, both principles of inheritance and molecular basis of inheritance. I will do it fully without any deletion of portion. I will do the entire thing. I will solve problems also. I will teach you how to remember, all that I will do. So apart from genetics, anything else that you want me to teach? Yes. Apart from genetics, so in class 12, any other topic that is tough for you? That you want me to put focus on, you want me to discuss questions on? No ma'am, nothing else. Fine. So do you like lecture based classes or question based classes? So do you want to discuss only questions, or do you want to have lecture wise theory based classes as well? So which type of classes would you prefer? So are you both giving term 1 exam of class 12? Yes. Amarnath, any other topic that is tough for you? Questionnaire, okay, fine. So from each chapter, is it fine if I discuss only the questionnaire so that it is easy for you? because in topics that is not actually needed to teach. For example, if you so genetics you find it tough, I will teach. So other than that, for other chapters I will bring only questionnaire rounds so that it is easy for you, so that we'll quickly finish it off. All right, yes, fine. So I just want to know what is your preference, what is that you expect from me. Yeah, that's fine. So you guys want me to teach genetics, okay? So next week if I start genetics, will you guys come? Yes. So 3 days I will teach the concept of so one day I will teach the concept of genetics, the next day we'll discuss questions. So we will go in that flow. Yes. Okay, you're from Tamil Nadu, all right. So you are not giving term 1 exam. Okay, fine. So I guess you're giving state board. Okay, done. So we will do it this way. So next week I will start genetics, probably say from Monday I will start genetics, because we are going to say goodbye to this chapter. So the next 2 days of the week we are just going to have mentees, so I have no plans. So Monday we will start off with genetics. So Monday if I start, Wednesday I will discuss the questions, and Friday I will take a lecture. Again Monday we will discuss questions. So I'll bring as much as questionnaire session as much as I can, okay? You have Kerala and you have term exams. So are you in CBSE? Are you in CBSE, or once again it is state board, Sneha? So if it's CBSE, then we'll have to focus on your term 1 as well. Yes, CBSE. Okay, so I will be discussing CBSE paper. So December 18th is bio paper. So if I discuss genetics, it will be easy for your term 1 as well. All right, done. So our plan is done for the next week. Fine. So let's finish off this chapter quickly. So in the last class, when we were talking about sexual reproduction in flowering plants, we were talking about double fertilization. So what is double fertilization and why it is called as double fertilization? So a quick recap of all that. Maybe we can discuss questions from your sexual reproduction in flowering plants also. So I will make up a schedule and I will share it with you, so you can see the thumbnail, so that you will understand when which class is there. So I guess day after tomorrow you have your first paper, that is English. So all the very best for your English exam. Fine. So let's move forward. So we were talking about double fertilization. So what is double fertilization? So once your pollen grain lands on the stigma, and if it's the right pollen grain, so then the process of fertilization proceeds, right? So the outer layer, so this is your pollen grain. So the outer layer of your pollen grain is your exine, and the inner layer of pollen grain is intine. So exine layer is made up of sporopollenin, and intine layer is made up of cellulose and pectin. So sporopollenin is what makes the pollen grain very resistant to high temperature, pH and acid. Okay. So now once the pollen grain is landing on the stigma, it can either be in the 2-cell stage. So the 2-cell means it can have your generative cell plus your vegetative cell. So if it's in the 2-cell stage, after it has landed on the right stigma, after the right pollination, it will enter the 3-cell stage. So 3-cell stage is when your generative cell undergoes mitotic division to form 2 male gametes. Plus you have the vegetative cell. So most of the angiosperms, they release the pollen grain at the 2-cell stage, and the remaining they release in the 3-cell stage. If they are released in the 2-cell stage, and they become the 3-cell stage, right? So now this male gamete has to fuse with the female gamete. So female gamete is present inside the embryo sac. So your embryo sac is 7-celled and 8 nucleate. Fine. So here you have 3 cells. Here you have 3 cells, and here you have 2 nuclei. So this end is called as the micropyle end, and this end is known as the chalazial end. Fine. So these 3 cells that is present in the chalazial end is called as antipodal cells. Fine. So they are antipodal cells, and these two are called as secondary polar nuclei. So these three structure together is called as the egg apparatus. So here these two cells are called as the synergid cells. Fine. And here you have what is called as the egg cell. So towards the micropyle end, you also have finger-like structures that is called as the filiform apparatus. Okay. Hello Dharani, yes, after a long time you are here. I hope you're doing well. So thank you so much for joining my session. You have sent me a doubt on Telegram. Yes, okay, I will get back to you after class. Okay, yes. So please don't forget to like, share and subscribe our session. So these finger-like structures are called as filiform apparatus. So now your pollen grain has to reach this egg apparatus. So now the pollen tube germinates to enter into the filiform apparatus so that it can reach the egg apparatus. Alright. So this is the pollen tube. So what helps in the germination of pollen tube is the germ pore. Fine. So this germ pore helps in the pollination of the pollen tube and it reaches the filiform apparatus. So once it reaches the filiform apparatus, so one of the male gamete, it fuses with the egg and this forms the zygote. So this is normal fertilization or normal fusion. So this is known as syngamy. But we have another male gamete as well. So the other male gamete, it fuses with your secondary polar nuclei and it forms the triploid endosperm. It forms the triploid endosperm nuclei. So since this is triploid, it is called as triple fusion. Since two fusions are happening, one is syngamy and one is triple fusion inside the embryo sac, so this process is called as double fertilization. Now this will develop into a zygote, this will develop into the primary endosperm nucleus, and this entire cell will be the primary endosperm cell. So this is what we were discussing in the previous lecture on your double fertilization. So now moving forward. Okay, so here, we now have two things, right? So now we have the zygote here. So the zygote is here. So here you have three nuclei fusing together. So this is your primary endosperm nucleus. So this entire thing is the primary endosperm cell. So now your process of fertilization is done. Now post-fertilization, what happens? So your post-fertilization includes your endosperm development first and then your zygote development. Right? So how does the endosperm develop? So this primary endosperm nucleus, it undergoes mitotic division and it forms many nuclei. So it forms thousands of nuclei. This is free nuclear division. So one example that we spoke is that your tender coconut water is the free nuclear endosperm and the white kernel is the cellular endosperm. So then later it forms the cell wall. So example, your tender coconut water, so this is your free nuclear endosperm and the white kernel surrounding your tender coconut, so that is your cellular endosperm. Fine, I hope this is clear. This part is clear. So after this, we went on to talk about what are endospermous seeds, what are non-endospermous seeds. So endospermous seeds is when the endosperm is retained till the seed maturation. So non-endospermous is when the seed is not retained till the seed maturation, when the embryo consumes it fully. So basically your endosperm consumes, your endosperm supplies food to the growing embryo, right? So that is how it is. So Prince Kumar, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So Prince, you also only come for class 12 topics. So are you in class 12? So what are the topics in class 12 that is tough for you? Please, you can put it, because I've been asking the others, because I want to know what you guys expect out of this session. Right? So after this, we started talking about the seed development. So how does the seed develop? Because the embryo gives rise to the seed, right? So now how does the seed develop? So you have the monocot seed and you have the dicot seed, right? So dicot is very simple. So you have two cotyledons like this. You have an embryonal axis. So the embryonal axis above the cotyledon is known as epicotyle, and the embryonal axis below is known as hypocotyle. Fine. So these are cotyledons. So your epicotyle gives rise to plumule. So plumule gives rise to shoot. So hypocotyle gives rise to radicle, and the radicle gives rise to root. So this is your dicot seed, and it is very easy. No, so you are a dropper. So have you taken a ear drop? Okay, fine. So now how about the monocot seed? So the monocot seed is slightly different. So you have the embryo here and you have the aileron layer like this. So you have the aileron layer. So you have the endosperm. So here you have the cotyledon. So the cotyledon is covered by a sheath that is known as scutellum. So here also you have cleoptile. So here also you have plumule. So this part, if it gives rise to the shoot, that is plumule. So this is surrounded by a sheath that is called as your cleoptile. So if this is your radicle, so your radicle is surrounded by a sheath that is known as cleorrhiza. Right? So with this is where we exactly stopped in the previous class. So now let's move forward. Okay, your first attempt was done in 2021. Fine. So which means now you're so have you scored good marks? Are you trying to give NEET once again? So where are the chapters that you felt very weak? So what are the weak chapters? What are the chapters that you want me to discuss? Yes. So guys, you can get in touch with me in the Telegram group. So beneath my video, there is a description, right? So beneath the description, there is a Telegram link. So please join the Telegram group, and from the Telegram group, you can find me. My name is Krishna. I'm one of the admins. So if you guys have any doubt, you can put it there. I will get back to you. Fine. So you've got 504. Wow, that's a good score. Very good, Prince. So you are expecting a good college. That's good. Fine. So, so what are your further plans? You're waiting for counseling? Yes? That's a very good score. So, many congratulations. Fine. So, now let's move forward. So, we are talking about the seeds. So, in angiosperm, so seeds are the final products of your sexual reproduction. The seed develops from the fertilized ovule. Okay, and the seeds are present inside the fruit. So we have the ovary and the ovule, right? The ovary develops into the fruit and the ovule develops into the seed. Fine. So the seed consists of seed coat, cotyledons and an embryonal axis. So cotyledons are simple structures which are swollen due to the storage of reserve food material. So seeds can be non-albuminous or ex-albuminous. When the endosperm is completely consumed, it is non-albuminous or ex-albuminous. It is non-endospermous as well. So when the endosperm is completely consumed by the developing seed, example pea and groundnut. So albuminous seed is when a part of endosperm remains till seed germination, that is albuminous or endospermous seeds, right? So example is your wheat, maize, barley and castor. So no, so you are trying to give another attempt of NEET, Prince? Preparing for NEET 2022 NEET exam. Very good. Okay. So I guess my lectures are of help to you. So Prince, can you tell me what are the chapters that you find weak? So do you expect a lecture session or do you expect only questionnaire sessions? Yes, ma'am. A very, very happy good evening. So thank you so much for joining my session today. So please do like, share and subscribe. Yeah, so this is the difference between albuminous and non-albuminous seeds. So occasionally in some seeds, so sometimes the new cellulose also remain. The remnants also remain, right? So even though you clean everything, some small portion is always left. Sometimes in certain plants, when the leftover portion is the new cellulose, so that is seen in case of your black pepper and beet. So Sneha, since you are preparing for term 1, so this question is always asked. So the remnant of the new cellulose is seen in your black pepper and beet, and this remnant is known as your perisperm. So perisperm is when your new cellulose persists in the seed condition. It has remained, okay? That is your perisperm condition, alright? So integuments of the ovules harden and they become rough protective seeds. So integuments are the coverings which cover your new cellulose, and inside the new cellulose you have the megaspore mother cell, right? So this integuments, they develop and form protective seed coat. So the micropyle remains as a small hole or the small pore inside your seed coat. So that is the way by which your seeds get or exchange gases. So it facilitates the entry of oxygen and water during seed germination. So in the micropyle end, the ovule is not covered, right? So this is my ovule. So I have the integuments, outer and inner integuments. So this region, it is not covered. So this is my micropyle region. This persists even in the seed formation because that is how the oxygen enter. That is how they facilitate the exchange of gases. So when the seed matures, it loses its water content and it becomes dry. So if at all the seed is facing unfavorable conditions, then in that case the seed will become dormant. So if the conditions are not favorable, there is no point in the seed germinating. It's anyways going to die. So the seed as it becomes very dry, it also can reduce its metabolic activity and it can enter a dormant stage. So when the conditions are favorable, they will germinate to form a new plant, right? Yeah, so this is what I told you. So moving further. So further, the ovules develop into seeds and the ovary into fruit. So this proceeds simultaneously. So this does not happen one after the other, just like your endosperm and the embryo, but this happens simultaneously. So your ovules develop into seed, at the same time your ovary develops into the fruit. So the wall of the ovary develops into fruit wall, that is pericarp. So the ovary develops into the fruit wall. So there is a wall of the ovary, so that develops into the fruit, right? So now it has a wall. So every fruit has a skin, apple has a skin, banana has a skin. So that is my ovary wall. So that fruit wall, now it has become the fruit. So it is called as a fruit wall. So the fruit wall is called as the pericarp now. Okay. So fruits can be fleshy like a guava, mango and orange. It can be very dry like groundnut and mustard. Okay, so the fruits have evolved mechanism for the dispersal of seeds. For example, now if I have an apple in my hand, so now the apple and the apple tree knows how to disperse the seeds. Say for example, apple we consume. There are a lot of fruits which are left unplugged. Even then the plants know how to disperse the seeds. Only when it disperses the seeds, it is trying to sustain its survival, right? Because seeds are the way by which the plant survives. Right? Yeah. So fruits form the ovary, other floral parts degenerate and fall. So in a flower you have other floral parts like the petals, sepals, etc. So they degenerate and fall because now they have no role. But in apple, strawberry, cashew, the thalamus contributes to fruit formation, that is false fruit. So you have two types of fruit. So one is your true fruit. So true fruit is formed by the actual fertilization of the male and the female gamete. So true fruit is formed by the actual fertilization of the male and female gamete. Fine. So now you have what is called as false fruit. So in false fruit, I never said the fertilization will not happen. Yes, here also I have fertilization happening between the male and the female, but apart from that, the thalamus also contributes. So what is the thalamus? So thalamus is the platform from which all the 4 floral whorls of a flower develop. So we have 4 floral whorls, right? Andrecium, gynoecium, calyx and corolla. So they all develop from a point called as thalamus. So when I draw a flower like this, so here I have my petals, so here I have my sepals. So this is my thalamus. So when the thalamus also contributes in fertilization, that is false fruit. So when the fruits develop from the ovary, that is true fruit. So few fruits do not have fertilization at all. So here in false fruit, fertilization happens, but thalamus contributes. But in case of your parthenocarpic fruits, there is no fertilization at all. So example, banana, through the application of hormones, they give you more bananas. So seeds are advantageous to angiosperms because seeds are the end product, right? So why seeds are advantageous? Because it is the reproductive process, it undergoes pollination and fertilization, and it is independent of water, right? So seed formation is dependable, the seed can form anywhere. For example, I am carrying a mango from Kanyakumari to Kashmir. So I eat the mango and throw the seed there. Yes, if the conditions are favorable, the mango tree can grow there, right? So seeds have better adaptive strategies for dispersal to new habitats, help species to colonize in other area. Very simple. So I am carrying the fruit from Kanyakumari. So it is not that the seeds will not travel with me to Kashmir, right? It will travel, but in Kashmir it will see how it can develop better or how it can adapt better. Seeds are food reserves. They have food reserves. They have the ability to nourish themselves till they are able to photosynthesize. So the seed coat provides protection to young embryos. So sometimes the seeds are very hard, right? Even the mango seed is very hard. So every seed is hard. So seeds are products of sexual reproduction. So they generate new genetic combination and variation. And of course, seeds are the basis for agriculture. So if I want to have a crop plant, if I want to have a farmhouse, I ultimately need seeds, right? So these seeds can be preserved as well. So dehydration and dormancy of mature seed is crucial for storage. I can even store the seeds. So what is viability of seeds? So sometimes some seeds can stay for a long time. So at home you have dhania, right? So when you sow dhania, it gives you the coriander plant. So those dhania seeds are very viable for a long period of time. Say for example, I got the dhania seeds in the month of January and now it's December. Now if I sow it, yes, it will give me a plant. But there are some seeds which loses its viability very quickly within weeks or within few months itself, right? So viability of seeds varies from one plant to the other, right? So viability can be very small, so it can be only for 1 month. So it can be from weeks to months alone. Sometimes the viability of the seed can be very long. Maybe the seed is preserved for 2000 years. It can still give rise to a new plant. So in case of an archaeological excavation, so they found out one seed that was deep buried and they found out that the seed's age is around 2500 years, and when they planted it, surprisingly the seed was able to germinate. So the viability so the viability of that seed was able to be maintained, right? So that is viability of seeds. So now moving to two other conditions, one is apomixes and the other one is polyembryony. So what is apomixes? So apomixes is again formed without fertilization. So when you have, so apomixes is when you do not have fertilization. So here no fertilization happens. Okay, then how is the fruit formed? So the fruit is formed when the new cell participates in the process of fruit formation, fruit or seed formation. So once again, so you have true fruit, so where you have proper fertilization between the male and the female gamete, okay. So then you have false fruit. So false fruit is when the fertilization happens but thalamus contributes, right. So then you have parthenocarpic fruits. So in parthenocarpic fruits, there is no fertilization at all. So in apomixes also, there is no fertilization, but nucellus plays a role in forming the fruit, right. So apomixes is one such condition. Now what is polyembryony? So poly is many, embryony is embryo. So when you have many embryo, that is polyembryony. Yes, Shubham sir, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. So polyembryony, so poly means many, right. So when you have many embryo, so this is seen in the case of citrus and banana. So when one male gamete has the capability to fertilize more number of egg, that gives you polyembryony. So here you have many embryos developing. So this is polyembryony. Fine. So the last slide for today, so that is the types of ovule. So based on the arrangement of the funicle and the body of the ovule, you have many types of arrangement. Okay. Ma'am, one doubt rising in my mind. Yes, Mystic Mind. So I hope you have a doubt related to biology. And anyways, thank you so much for joining my session. So please do like, share and subscribe. Yes. So what is this? What are the different types of ovules based on their mode of or orientation of arrangement? So first we will talk about anotropous. So okay. So first is atropous. So atropous is when everything is in the straight line. Right. So when everything is in the straight line, that is 180 degree, that is atropous. So anotropous is when it is at 90 degree. Okay, so yeah, so that was a very weird doubt. So why can't we make our own food? So why are we dependent on green plants? Because green plants are autotrophs. So what if we can inject chlorophyll, and if we have sunlight, why can't we make our own food? Well, so we do not have, so even if you inject chloroplast inside your body, so for the process of photosynthesis, you need a lot of enzymes. So those enzymes are not able to synthesize in our body, because of which we cannot perform photosynthesis. So that is point number one. So even if you have all the enzymes, even if you have the chlorophyll, so a body condition is not suitable to perform photosynthesis, because our body temperature is 37 degree, right? So when plants are performing photosynthesis, their temperature is different. So according to the temperature, only enzymes can work. So the enzymes in our body have maximum efficiency at 37 degree, but the enzymes that you take from plants, it's not that they are active only at 37 degree, they can denature inside a body. So these are the reasons why we cannot become autotrophs, and we are still heterotrophs, and we are dependent on the plant. Okay, so amphitropous. So amphitropous is when the funicle is at 90 degree to the body. Hemianotropous. So anotropous, okay, so let's do this. Okay, so I guess I confused you. So let's go slowly. Fine. So now anatropous. So anatropous is the straight line. So anatropous is 180 degree. So hemianotropous means 90 degree. Right? So here camphilotropous means when it is curved. So circinotropous means when it is circled. So amphitropous means it is inverted. So atropous means it is in the straight line, it is facing the top. So these are the different types of ovules based on their orientation. So I guess with this we are done with this chapter. So we say a goodbye to this chapter of sexual reproduction in flowering plants. So, so from your suggestions, I will make changes to my schedule so that you benefit most out of the session. And for all of you, so there's a mentee tomorrow, and it's a gentle reminder. So please join me for the mentee at 5:15 tomorrow. So I want to see more number of participants. So more the number of participants, more interesting the mentee will be. So please join me for the same. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So thank you so much for joining my session. So I'll meet you tomorrow in the mentee. So until then, this is me signing off. So take care and bye.